This method may help with optimizing and selecting clinically useful immunotherapies for Clostridium difficile infection in a patient-specific manner and elucidating humoral immune responses to Clostridium difficile.
The main advantage of this technique is that it enables accurate and simultaneous quantification of multi-isotype and strain-specific antibody responses to C. difficile antigens. Generally, individuals new to this method will struggle because expertise is required in establishing and optimizing a panel of highly purified antigens and setting up the technological platform. Visual demonstration of this method is critical, as methodological steps involved in preparing a microarray plate, arrays printing, assay procedure, and data analysis require attention to the details. Demonstrating some of the procedure will be by Sam Greenwood, a technician from my laboratory.
To begin, dilute Clostridium difficile antigens in printing buffer to optimum concentration. Next, transfer 10 microliters of antigen solution to a 384 well plate. Cover the plate with a plate seal, and centrifuge at 300 times gravity for 5 minutes at room temperature. For microarray printing, use a handheld gas burner to heat the silicon pin 3 times for 2 seconds each. Then, rinse the silicon pin in clean water 3 times for 3 seconds each. Place the microarray plate in the loading cartridge of an arrayer. Eject the tray